Right. So we'll be discussing the class test paper. Question number 1. So a small tube called the catheter can be inserted into the blood system through the vein, and it can be threaded through the vein into and through the heart until its tip is in the pulmonary artery. So this is usually what the doctor will do, right? Among all the blood vessels that you've learned, artery, vein, capillary, right? Which one got larger diameter in the lumen? So vein is the one with larger lumen, right? So that's why they want to put the catheter into the vein. Catheter is a flexible tube, okay? So you put it into the vein and follow the vein and move all the way until it comes to the heart, which is actually from vena cava goes to the right atrium and then into the right ventricle and then go to the pulmonary artery. So they say that the end of this particular catheter has got a balloon, and this balloon can be used to measure the pressure changes in the pulmonary artery. So this figure 1.1 shows a section to the heart with the catheter in place, and graph 1.1 shows the pressure changes recorded in the pulmonary artery by this balloon here. Okay, so this is the pressure changes in kilopascal in different timing in second. So A, name the structure of the heart P. So P is the septum. Okay, so P is the septum. Okay, B, complete the table below by placing a tick in the appropriate box or boxes to show which of the valves 1 to 4 will be open and which will be closed. So this is very straightforward, right? At the time X in the graph. So X in the graph, there's a sudden increase in pressure in the pulmonary artery. That means to say that ventricle contracts, right? It's the ventricle contracts that causes a sudden surge of blood into the artery. So sudden increase in blood. So you already learned in theory that two factors that affect the pressure, either is in the chamber or in the blood vessels, right? So can you tell me what are the two factors that affect the pressure in the chamber or in the blood pressure? Jia Ying?
Diastole and systole.
Systole and what?
Diastole.
Systole and diastole. Okay, what else? Second one is what? Jiang, open to the group. Open to the group. Volume of blood, yes, correct. Low cesium, volume of blood. Low cesium. Okay, it's the volume of blood. So you must remember, if you want to know a graph, which is graph of pressure versus time, so you need to know what affects the pressure, right? And the pressure, you learn the pressure in the 4 chambers, as well as pressure in the aorta, as well as pulmonary artery. So these are the two factors that affect the pressure. So you need to remember. So it's easy for you to find out, okay, what causes the pressure to increase here? Okay, and what causes the pressure to decrease here? Pressure is decreasing here and pressure is increasing here. So increase is because of the ventricular systole. Decrease is because the blood flows away from here. Because when the blood comes out, right, it passes here and flows away. So blood volume drops. So that's why the volume here drops. Okay, so question asks the student in B, complete the table below what happened in the region X. So region X is when the ventricle contracts. So when the ventricle contracts, we expect AV valve to close. So number 1 will close, number 2 will open, number 3 will close, number 4 will open. Okay, so therefore the answer for this is that number 1 will close, number 3 close, number 2 open, number 4 open. Correct? Okay, 2 and 4 open, correct. So 2 and 4 open. Okay, so that's the answer, right? Okay. Next question, C. Blood pressure is usually measured by placing a cuff over an artery in the arm. So usually when you measure blood pressure, usually it be reported as 2 pressures, right? So the normal pressure is usually, they say average normal is 120 to 80, isn't it? Both of them are millimeter mercury. All right. So there are 2 readings there. So the one on top is called the systolic blood pressure, and the one at the bottom is diastolic blood pressure. So this is measured in the artery. So that means to say that when they put the cuff over the artery and measure the changes in pressure, there'll be two pressure that will be measured, right? Systolic, diastolic blood pressure. So the question here asks a student, explain why the blood pressure in the left ventricle falls to 0 in the cardiac cycle. So that means you need to explain why blood pressure in the left ventricle can go all the way to 0, but the lowest blood pressure in the artery is actually 10 kilopascal rather than 0. So that means for the 3 marks that they give to you, your 3 marks must include explaining why blood pressure in ventricle goes to 0, and also blood pressure in artery lowest is 10 kilopascal and not 0. Okay, so first you need to explain about blood pressure. Why does it go to 0? Blood pressure goes to 0 it has to do with pressure depends on systole or diastole, right? So this is the first thing. And then pressure also depends on blood volume. So there are 2 things here for you to mention. Blood pressure in left ventricle falls to 0 because ventricle is undergoing diastole, right? It means it's not contracting, it's relaxing. So it's not squeezing to the chamber, as well as all the blood has been emptied into the arteries during ventricular systole. So when ventricular diastole happens, diastole, okay, then there is no blood inside, and then the muscles relax, okay? So therefore, the pressure falls to 0. So that is for the first marking point. So first marking point, you need to talk about, where is it? Okay. So you need to talk about the pressure drop to 0 because all the blood has been expelled from ventricle. So it is talking about the volume as well as and ventricular diastole. So there are 2 things you need to say. You have to talk about blood volume, which is 0 already, and the ventricle is undergoing diastole. So these 2 must be there to score 1 mark because these 2 affects the overall pressure in the chamber. Okay, so this is for this first part. Second part is, how come the lowest pressure in the artery is 10 kilopascal and not 0? Now, this is because of elastic recoil. So we know that in the artery, if this is artery, the blood is being pumped out from the heart beat by beat. Okay, that means pulses by pulses. So when the ventricle contracts, so a certain volume of the blood flows out from the heart, and then ventricle relaxes, right? Followed by the next ventricular systole, which pump another volume of, or another pulse of blood out. So you got 2 pulses of blood here, right? In between the 2 pulses in between them is empty. Okay, this part here is empty. So they're saying that how come this part is empty, right? There's a certain pressure is 10 kilopascal. Now you know that when the blood flows into the artery, right, the blood has got a certain pressure there. So the pressure of the blood will actually push the wall of the artery to stretch outwards. So actually the wall will stretch outwards as the blood flow into the artery, right? So this stretching is very important because it will reduce the pressure of the blood on the wall so that the wall will not burst, okay? So when the blood flows in, because there is elastic fiber in the wall, okay, so therefore the elastic fiber will be able to stretch when the blood flows into the lumen. So it will stretch, okay? The blood pressure will push against the wall and the elastic fiber will stretch. And then as the blood continue to flow away from this artery, this elastic fiber in the wall that has been stretched will undergo elastic recoil. So elastic recoil will occur as the blood flows away, right? Because of the potential energy. So this is called elastic recoil. Is a biological term. You have to write the whole term together. Okay, so this pulse of blood is flowing away, and as it flow away, elastic recoil occur. So when elastic recoil occur, it will push this pulse of blood, And when it flows away, it will actually push into the space, the empty space already, because when the blood flows away, it will be replaced by the empty space. So it push into the empty space already. So therefore, this pushing due to elastic recoil created the 10 kilopascal of pressure in the lumen of the artery between the two pulses. So therefore, the lowest pressure in the artery is 10 kilopascal and not 0 because of the elastic recoil of the elastic fiber, okay, in the tunica media of the artery. So this elastic recoil is able to maintain the blood pressure between the two pulses of blood. So it's not 0 because of the elastic recoil. It created a 10 kilopascal pressure inside here. So it maintains the blood pressure between pulses, and hence it says that it smoothened out the blood flow. That means it's not suddenly you got a blood pressure suddenly 0. At least it doesn't go back to 0. So it smoothened out the blood flow. Okay? Because this pressure is also able to push the blood forward. As the blood flow away, the wall recoil back and pushes the blood forward. So smoothen the blood flow between pulses. Okay, this one is ignore. That means if you talk about smooth muscle contraction, we don't give mark because this pressure 10 kilopascal is not because of smooth muscle, it's because of elastic fiber. Okay, so this one you have to understand. You have to understand this one clearly. Right? So next question, number 2. So there's a graph given to show you the pressure changes in the 4 chambers of mammalian heart. So on top here is about left side of the heart. So you've got left ventricle and left atrium. In the middle here, this graph is about the right side of the heart. So right atrium, right ventricle. At the bottom here is the volume in the ventricle. So I'll say that this is the blood volume. Okay, blood volume ventricle. So this graph is a very accurate graph. Okay. This graph is a very accurate graph because I think this is from our old textbook, which is very, very concise and precise. The old textbook, not the previous version. It's a very, very old version, which is very concise and precise. Okay, so this graph is the best. So you can use this graph for revision as well. So let's look at the question. A, state the term used for each of the period A and B. So period A starts from here. That means this is when ventricle contracts, right? Okay, and it ends here. So you see, after this point, these 2 lines separate already. So when these 2 lines separate, it means that the pressure in the aorta and the pressure in ventricle, they are separated. That means the valve between them is closed already. Okay, so that means ventricles start to relax from this point onwards. Right? Ventricle starts to contract at this point onward. So A is ventricular systole. B, so starting from here when ventricle relaxes all the way until the next ventricular systole. So B is ventricular diastole. Okay. So check the spelling must be correct to score. If the spelling is wrong, cannot give mark. So check that the systole and diastole are both spelled correctly to score. Okay, whenever you're asked to name, when the spelling is wrong, there'll be no mark. Okay. So B, why must the blood pressure be higher in the left ventricles than in the right one? 2 marks. So check how to write to score the full marks. Now, left ventricle pumps the blood to the whole body. If you don't write whole body, I will also accept you write systemic circulation. Okay, but you also must say that this systemic circulation is very far or long distance. So this is the first marking point. That means you must tell me left ventricle pumps the blood to where? And then this location is far away from the heart, so therefore it needs a higher pressure. So for right ventricle, it pumps the blood to the lung, or you can also write as pulmonary circulation. But you also must say that this pulmonary circulation is near to the heart or it's a shorter distance away from the heart. So that's why we need lower pressure. Okay, so both location and distance must be mentioned to qualify for 1 mark for each of the ventricles. Anything missing, no mark. Okay, if the students talk about go to the whole body, go to the lung, but did not talk about distance at all, so overall 0 mark. Okay, because it doesn't qualify for why is it the pressure is higher. It must be the longer distance that pressure is higher. Okay. Okay, next question is C. Explain why, what happens in the heart when the pressure in the left ventricle exceeds that in the aorta. So this is very, very easy, right? So when the pressure in the left ventricle exceeds that of aorta, of course, the semilunar valve will open and then the blood will flow from the left ventricle to the aorta. Okay, so let's check the mark scheme. Semilunar valves open, blood will be expelled or blood will move, okay, blood will flow, blood will be pushed from the heart or the left ventricle into the aorta or systemic circulation. Okay?
Teacher.
Yes.
Is blood propelled into aorta accepted?
Blood is propelled into aorta is accepted. Yes, it's this marking point. Expel or push or propel or whatever. Okay?
Okay, thank you,
Or
teacher.
flow into also can. All right, part 2. Part 2. Explain how the backflow from aorta is prevented as the pressure in the left ventricle falls. 1 mark. Easy, right? Closing of the semilunar valves. Okay? All right, so D. Explain the small surge of pressure in the right atrium and right ventricle at X. So let's look at X. So this is X here. So it says that there's a small surge of the blood pressure in the, this one is what? The ventricle. Okay, the solid line is ventricle and the dashed line is the atrium. So explain why this can happen. So you already know that this is the point where both the lines becomes the same, right? So when both the line becomes the same, that means both of them are connected. So that means when the pressure in the ventricle go below the pressure in atrium, right? The AV valve will open, so both of them are connected. So both of them got the same blood pressure already. Then as the blood continue to flow from atrium to ventricle, so the pressure will drop because of blood flows out from atrium to ventricle. Okay, and then the blood volume start to increase as it collects. Then over here, the sudden surge is because atrium contracts to squeeze the remainder 30% of the blood. Okay, the 70% flows in passively. 30% flow in because it's being squeezed by atrial systole. So the answer for D, why is there a small surge? Is because of atrial systole. So this is the name of the process. You always give the name, you get 1 mark. Okay, the name of the process is atrial systole. And then what happened? So because of atrial systole, the pressure in the right atrium increases. Okay, and then it pushes the blood or the blood flow from right atrium to right ventricle. Underline means the student must write right atrium to right ventricle. Okay, if the student write atrium to ventricle, no mark, because the question is asking about the right side of the heart. Okay, the graph there in X is actually showing the right side of the heart only. So this is X, this is the left. So therefore you need to specify according to the question to score. Okay, so this is something that you got to be very careful. You can score the mark, but you got to be very careful and very detailed. Okay. Anna? All right, question 3. So question 3 is a capillary. So A, explain how tissue fluid is formed from the blood plasma, 3 marks. So that means you need to describe the process of formation of tissue fluid from blood plasma. and also give the name of this process. Every time there's a name, you write the name, it's only one word, you already get 1 mark. So it's best to start with the name of the process and then elaborate. Okay, so let's look at the mark scheme. So say that the blood plasma, okay, the formation of tissue fluid from blood plasma is by ultrafiltration. So 1 word, 1 mark. Okay, ultrafiltration means filtration under high pressure through a filter. Okay, so then you elaborate what is ultrafiltration. So there's high or higher blood pressure or hydrostatic pressure at the arterial end of the capillary. Okay, so this pressure will force the blood plasma to flow through the pores or gap, which is found on the endothelial wall, which is the capillary wall, or this gap is found between the endothelial cells. So this blood pressure will force the blood plasma to pass through the pores or the gap between the endothelial cells of the capillary wall. So what are the things that can pass through are actually water and solute molecules with a size less than 96,000 molecular weight. So they will be able to pass through. So I prefer you to actually write this answer than 10 nanometer. Okay, 96,000 is a better answer than 10 nanometer. Okay, Ken? So this is how tissue fluid is formed from the blood plasma. Okay. So shall we continue next question? Okay, next question is B. So B, some of the tissue fluid does not return to the capillaries, but enters another set of vessels. So name the fluid in another set of vessels. So the other set of vessel, right, is actually blind-ended. Right, so you got a capillary flowing here, right? Okay, arterial end and venous end. Then you've got your tissue here with all the cells, isn't it? Okay, tissues and all the cells here. So you got out of 100% of the fluid that comes out from blood plasma, 90% will flow back at the venous end of capillary, and then you got 10% will flow into this vessel, which is blind-ended vessel. This is a lymphatic vessel. So they ask you for the name of the fluid. So the name of the fluid is actually the lymph, L-Y-M-P-H, or lymphatic fluid. Okay, so C, describe the composition of the fluid that drains into another vessel. So what is the composition of the fluid here? So then you must know what happened when the fluid from blood plasma goes into the tissue fluid. So all the useful materials will be taken up by the cells, so the oxygen, okay, and the nutrients will be taken up by the cell. Not all, okay. Most of them will be taken up. Okay, it is by diffusion until equilibrium is reached, isn't it? Okay, unless it's active transport. If diffusion, then after they take, the concentration on both sides will be the same, right? So of course the cell will use oxygen for respiration, and therefore it will release CO2, right, as a product of respiration, as well as it will release water is also the product of respiration, right? Okay. And of course, there are cells like cells in the liver, okay, and that they will actually produce urea from excess amino acid. So urea will be produced, which is a waste material. And then there could be some white blood cell that will secrete antibodies, right? And it could be, if you say endocrine gland, so this tissue can be belong to tissue in endocrine gland, so they can be secreting hormones, right? So therefore, these are the things that be secreted into the tissue fluid. So what will be the composition of the tissue fluid? So it may contain metabolic waste, isn't it? Metabolic waste, such as CO2, carbon dioxide, water, okay, from respiration. So CO2 and water from respiration, maybe nitrogenous waste, the urea, okay. It may be containing hormones that are secreted. It may be containing proteins such as secreted by the cell, okay, such as antibodies. Certain hormones also are protein, okay. It can be also nutrients that are secreted by the cell, because don't forget, liver store glycogen, right? So therefore, when your muscle cells need more glucose, they may cause liver cell to hydrolyze glycogen into glucose, and then glucose will then diffuse into tissue fluid, then go back to the bloodstream, right? Transported to the muscle cells that need it. So there will also be nutrients as well. Okay, so therefore, let's look at how to give the marks. Let's look at the marking scheme. So metabolic waste is actually 1 mark. So you got the waste, water, nitrogenous waste, CO2 is 1 mark. Any useful substances secreted by the cell, hormones, proteins, that proteins is antibody, for example, okay. Nutrients, okay, nutrients, because it is being hydrolyzed from the storage organ like adipose tissue, right? The fat, triglycerides will be hydrolyzed into fatty acid and glycerol, right? And then glycogen hydrolyzed to glucose. So this one will be the nutrients will be mobilized. So from the storage organ to the tissues that need those nutrients, okay? I will not give mark for this one. This one is not relevant. Okay, so I only give mark for first 3 marking points. What is there rather than what is not there. I only give mark to what is there rather than what is not there. So that's the question asking for what is there, right? So I'm not interested in what is not there. Okay. So D, explain why large multicellular animals need blood circulatory system. Blood circulatory system. So you already learned that as the organism from unicellular becomes multicellular, so the size will increase, right? You learned that the size will increase. And when the size increases, if I divide this into the size of individual cells, then you start to see that a lot of cells now is no longer directly exposed to the external environment for direct exchange of material with external environment. Okay, let's say for example, this multicellular organism, big, so what's the problem? So big, the problem is when the cell is multicellular, it's big, right? Big, so there's got lower surface area to volume ratio. So this is what all of you have learned, right? So what problem do you have here now? Lower surface area to volume ratio. In this case, a lot of cells are far away. So cells inside the body are far from the exchange surfaces. So exchange surfaces means this layer here, right? I hope I can draw it nicely, but I don't know whether I can or not. Okay. Okay, this external layer, okay, how? Okay, this external layer, okay. So many cells are far from exchange surfaces. Exchange surfaces is where you can get oxygen that you want. You can get the nutrients that you want. Also, you can remove the waste. Okay, this is called exchange surfaces. Exchange between external and internal environment. So because the cells are very far away, if you allow material to move between external and internal environment, let's say between this cell and the external environment. So you want oxygen and nutrients to go in, you want the waste to go out, okay? So the diffusion distance becomes very far. So cells inside the body are very far from exchange surfaces, which means to say that the diffusion distance, which is represented by the symbol L, is very far. So which means to say that transport of material to the cell by diffusion alone is very slow, right? Because the distance is far, so diffusion rate becomes very slow. So diffusion alone is inefficient to get enough exchange materials between external and internal environment. So therefore, we need to have blood circulatory system whereby it is able to, now blood circulatory system, what does it do? It's got a heart that pumps. So when it pumps, it creates a high pressure. And this high pressure actually pushes the blood to flow. So the blood flow is not because of diffusion now already. The blood flow because there's a force that creates a hydrostatic pressure that pumps the blood around the body. So the movement becomes very fast because of the pump. So that overcomes the problem where the cells are very far from exchange surfaces. No problem. Because I got a highway, I can send the materials at high speed to every part of the body because of the pump in the blood circulatory system. Okay, so this one solves the problem of far away from exchange surface. Okay, and secondly, it allows, so this is a high speed because of the pump. So first function of the blood circulatory system is the heart pumping will create the high pressure to push the blood to move very quickly from exchange surfaces to the cell and from cell to exchange surfaces. Okay, so high speed. Second is large quantity. Large quantity can be transported because you got a special blood vessel where all the exchange materials will flow within the lumen of the blood vessel. So it's a highway. You got a special place for those materials to be transported, so you can transport large quantity at the same time, as compared to the molecules have to move by itself by diffusion, okay, from one cell to another cell, so it's very slow. But this one, it doesn't move through the cell, it moves through a hollow lumen, you know, so with very minimal resistance to flow. So large quantity can move. Okay, so let's look at the mark schema. I explained for you to understand. Okay, and now is what are the points that you're looking for to score. So because of multicellular organism, it has got small surface area to volume ratio. So what's the problem? The distance between the internal and external environment is very far. Okay, is very far. So this is the first problem. So therefore, to allow material to move between internal and external environment by diffusion alone is too slow. Okay, or you can say that the blood circulatory system will increase the rate of transport, or the heart will pump to create a pressure or hydrostatic pressure to push the blood to flow around the body in the blood vessel. So that will increase the speed of flow compared to diffusion alone. Okay, something to that meaning you'll get a mark already. That means the speed of flow is increased because of the pumping of the heart. Okay, and then this one, number 3, is actually large quantity. So I will say large quantity is a keyword. Large quantity of what? Okay, large quantity can be transported. So you can say oxygen or nutrient, CO2, urea, water, etc. can be transported inside the blood vessel or in the blood. Okay? Okay. So next question. Question 4. Explain what is meant by the term transpiration, 2 marks. So this is definition. So just straight away, the definition will do. Transpiration is the loss of water vapor. If you don't have vapor, no mark. Okay, loss of water vapor from the surface of plants. Okay, of course, if you write from the stomata also can, because 90% of water loss from the plant is through the stomata. So you can write stomata also, I will accept. Or you can say that from surfaces of plants also can. Okay. If you say by evaporation and diffusion, I will accept. If you say by evaporation, I will not accept. Okay. So I'll accept. Another point is by evaporation and diffusion. That one I accept. Okay. Right, so the next question. So the next question, there's a graph that's given there to show you y-axis is the water absorbed or transpired in gram. Right, so you've got 2 lines here, right? So solid line is water being absorbed by the plant, dashed line is water being transpired by the plant, and x-axis is the timing in 24 hours of the day. That means 12 o'clock means it's an afternoon. So this is afternoon. So afternoon is when the sun is the highest on the sky, so it's very hot, so you can expect transpiration rate is the highest. So there's more water that is being transpired than being absorbed. Okay. Right. So let's look at the question. With reference to figure 4.1, part 1 suggests in terms of transpiration and absorption. Now, pay attention to the question. Your answer must be in terms of transpiration and absorption. It means your answer must write transpiration and absorption. Okay. When will wilting be most likely to occur in this investigation? So, of course, the plant will wilt when the amount of water absorbed, okay, is actually less than the amount of water that's being lost by transpiration. So therefore, the answer for this question is when. Okay, let's look at the mark scheme. So the answer is when transpiration is higher than absorption. So that's the answer, because the question asked for the student in terms of transpiration absorption. So therefore, of course, from the graph, it is between 4 hours to 16 hours, but you write this one, you don't get the mark because the question asks you to write about in terms of transpiration absorption. Okay, so let's check, is it true from 4 to 16 hours? Okay, from 4 to 16 hours. So this is a point here. So this is a point when the rate of transpiration is higher than the rate of water absorption all the way until this point. So which is actually 4 to 16 hours, right? Okay. But marks is only given if you say when water transpired is more than water absorbed. Okay. Part 2. Explain in terms of water potential. So again, you see here, in terms of water potential, when the plant cells will no longer be turgid and wilting may occur. So that means the plant cell loses, the plant cells loss its water. So how come water can move out from the cell? It must be driven by water potential gradient, right? So which means to say that the water potential of the cell, right, must be lower than the water potential of the surrounding, right? So water potential of the cell is lower than the water potential of the surrounding. So this is cell here. And then this is maybe the environment. Okay. So that, no, sorry. I put it the wrong way already. Sorry, this is wrong. Okay, the other way around. Okay, like this. Okay. So when the water potential of the cell is higher than water potential environment, so it will cause the water to move out the cell down water potential gradient by osmosis, right? So down water potential gradient, okay, by osmosis. So this is when the plant cell will become no longer turgid, and then the plant will wilt. So the cell is no longer turgid, the plant will wilt. Okay, so that is answering the question using water potential. Okay, so let's check again the mark scheme. So in the mark scheme, so when will this happen? When the water potential in the atmosphere is less than water potential in the cell. Okay, or the water potential environment less than that in the cell. So water will move down water potential gradient. Okay, or from a region of higher to lower water potential, or you leave the cell. So that's all, 2 marks. Where's my osmosis? Okay. By osmosis, where's my by osmosis? Not there. Where is it? Where? Here, okay. So, how come I want to type and then you go away? Okay, hang on. By osmosis, okay. Comma, by osmosis. Okay. Yeah. So you need to write by osmosis. Okay. First, talk about the differences in water potential. Second, talk about movement of water down water potential gradient. Third, I think I'll put this as a separate point. Okay, I put this as a separate point so that you will get the mark. Okay, by osmosis. But I can actually put this as part of the second marking point. That means if you don't write osmosis, even though all these are correct, you will not get the mark. So just to remind yourself that always remember to write osmosis. Always remember to write, okay, the full package of answer for this kind of question. Okay, right, so next question. So this is a stem. So the diagram illustrated that a ring, a complete ring of the bark was removed from the stem. So this region is called the ring, the area of the bark that's being ringed, means removed. So this area of the bark happened to be at the phloem region. So the entire phloem in the stem have been removed. Okay. So the concentration of sucrose was estimated in the stem region immediately above the ring and the stem region and the roots below the ring. So the concentration of sucrose was also estimated from the same region in the unringed plant. So you got a ring plant, right, which is the experiment over here, and the data is here. And then this is a control. Control means it's a normal plant where you don't ring it. So you want to compare between the control and experiment, what's the difference? And then these are the 3 different locations where they measure the sucrose for the plant. So you got a stem above the ring, the stem below the ring, and also the root, which is below the ring. Okay, so you can see that for normal plant, we expect the sucrose to be flowing from above the ring to below the ring, and from here it flows to the root, and from the root, this will be taken up by the cells in the root in this case here. Okay, cells in the root, okay, which is considered as the sink, and they'll be taken up for 3 processes: respiration, growth, and storage. So please remember, so these are the use of the uses of the sucrose in the sink. Okay, in the sink. Respiration, of course, all cells require respiration. So you use the sucrose, hydrolyzed to glucose and fructose, use the respiration to produce ATP. Okay, growth. Of course, ATP can be used for growth, and also the glucose, okay, can be converted to cellulose. So this is used to make new plant cell, right? So during growth, there's cell division and a cell elongation, right? So during cell elongation is when the cellulose wall has to be produced, right? So storage is you store sucrose converted to glucose and fructose and then converted to starch. So all this happens in the cell in the sink. So that's why you see that this is 0.3. So which is lower than 0.41, because once it reaches the sink, the cell will take away the sucrose to be used for all these processes, while the new one is being transported in. So the rate of uptake is faster than the rate of transportation. That's why it's 0.30. Okay, it's like the rate of you taking out money from the bank is equal to the rate of your father putting in. The amount of money in the bank is still the same. So now we have got sucrose flowing in, right, from the stem, right? But we also got sucrose flowing out. So the sucrose flowing in is slower than the slower. Okay, is it slower? No. It's not slower. It's faster than the rate that the rate of the sucrose taking out. So that's why you still got 0.30 there. You still got 0.30 because rate of transporting in is faster than rate of transporting out. Correct. Rate of transporting out slightly faster than rate of transporting in. Okay, rate of transporting out slightly faster than rate transporting in. Okay. All right, so let's look at the question A. Or let's look at this data first. Above the ring is 0.60. Okay, but in a normal plant is 0.43 because it can flow from here to here. But now after you ring it, it cannot flow from here to here anymore because there's no phloem that connects between upper part of the ring and lower part of the ring, which is here. Right? So from here to here, there's no phloem. So whatever sucrose that comes here will actually collect over here. And then there's no more sucrose to come in from here to here. So whatever sucrose that flow downwards, finally, there's no sucrose left here. Why no sucrose left here? Because there's no input, but there is output. That means no more money come in, but money go out. So overall becomes 0. Okay. So every time you want to talk about change of concentration of something, you must consider both the input and output. Okay. No input, but there's output. The sucrose continue to flow downwards to the root. So until this region got 0 sucrose. Okay. In the root is 0.03 unit of sucrose compared to 0.30. That means there is uptake, okay, uptake of the sucrose from the roots to the cells, but there's no replenishment. So there is output, there's no input. Okay, sucrose is taken out, but sucrose is not replaced. So there's output, no input. So overall, the concentration will drop. Okay, so just think of every time you want to describe concentration, talk about output and talk about input. Okay, consider both directions. Okay, so let's look at the question A. With reference to the figure 5.1 and table 5.1, explain the result shown. So explain everything here, right? Okay, let's look at the mark schema. So ringing remove the phloem. So because of that, sucrose cannot flow down the stem. Okay, because of that, sucrose above the ring cannot flow to the stem below the ring. That means it cannot flow to the stem, flow down the stem, or cannot flow into the roots. Okay, so because of that, sucrose accumulate above the ring, whereas sucrose below the ring will flow to the root and being used by the root cells for respiration, for growth, for storage. Okay, so accept respiration or growth, okay, and or storage. Okay, all right, so next question B. Explain how mass flow of sucrose occurs in plant. Mass flow. Okay, how mass flow of sucrose occurs in plant. So we know that mass flow actually occurs in the sieve tube, right? So therefore, based on this question, you must always start answering from the question itself. It's talking about mass flow, and mass flow occurs in sieve tube. So you need to start answering from sieve tube. So why is there a mass flow in a sieve tube? Now, the meaning of mass flow, all the solutes and solvent molecules move in the same direction. So this is the meaning of mass flow. Remember, mass flow means the solute and solvent molecules are moved together in the same direction, which is different from diffusion. So when you talk about diffusion, diffusion of molecules is due to kinetic energy of the molecule. So if these are the water molecules, okay, in the beaker, okay, let's say these 3 are the water molecules in the beaker, they can be diffusing in different direction, you know. When you say diffusion, they can be all moving in a different direction, and this is considered as diffusion. But the moment you say mass flow means that all of them must move together in the same direction. Okay, all of them move together in the same direction. So that is called the mass flow. So mass flow in sieve tube and mass flow in xylem vessel, both are driven by certain kind of forces. Okay, so they're asking you for the forces that drive the mass flow in sieve tube. Of course, the mass flow in the xylem vessel is due to cohesion tension, right, which is transpirational pull, and as well as capillary pull, and there's also root pressure, right? 3 mechanisms. And then in the sieve tube is because of the pressure gradient. So this mass flow is due to the hydrostatic pressure, right? Pressure gradient, okay, which is higher in the source and lower in the sink. So that is the force. the very force that causes mass flow. So this is how you need to answer your question. Okay, you cannot start from companion cell H plus being pumped out. No, because the question didn't ask you about companion cell. The question asked you about mass flow in the sieve tube. So you have to start from sieve tube to make sure you score full marks in exam. Okay, so then from there, so the mass flow is because of hydrostatic pressure gradient, right? I'm answering the question. Now, one step backwards. Why is there a hydrostatic pressure gradient? Okay, so now I'm going to explain. The mass flow is because of hydrostatic pressure gradient. The hydrostatic pressure gradient is because of sucrose being loaded from the companion cell into the sieve tube via plasmodesmata in the source. So this is called crystal clear answer, right? Sucrose is being loaded from companion cells into the sieve tubes via plasmodesmata or through plasmodesmata in the source. Okay, so this loading will result in next thing. So what happened after the sucrose is loaded? So the water potential in the sieve tube will increase. Increase, right? No, decrease. Decrease, will decrease, right? You add solute, so you will reduce water potential. So this will cause water to move into the sieve tube from the surrounding cells, okay? Move into the sieve tube is actually by osmosis. Okay, by osmosis, because sieve tube element is a cell that is alive, so there's membrane. In order to enter the lumen, it must pass through the cell surface membrane, so it's by osmosis. Okay, so the movement of water into the cell will cause increase in hydrostatic pressure at the source. Okay, so that's why it drives the mass flow of the sucrose. Of course, you can talk about sucrose unloaded in the sink, right? Water follows. So therefore, lower hydrostatic pressure in the sink. So this to explain why is it the source got high hydrostatic pressure, the sink has got lower hydrostatic pressure, so overall there's hydrostatic pressure gradient. So that's what the question is asking for. So if you tell me a lot of story about the loading of sucrose into the companion cell, maximum mark you can get is under AVP, which is actually 1 mark only, because it's too far away from what the question is asking for. Okay, so therefore you were telling me that H plus is actively pumped out from the companion cell, okay, and then the sucrose is co-transported with the H plus into the companion cell, okay, and then water enters the companion cell. All these are marked as 1 point only. No matter how many points you write here that's correct, you only get 1 point, because this one, the answers are too far away from what the question is asking for. So I hope that with this question, you actually learn exactly what you need to write to score full marks. You cannot just, oh, this is related to loading, so you write everything about loading. No, which part of loading? Because you want to score full marks. So you need to answer the question to score the full marks. Okay, so I hope that with this question, you actually learn exactly what you need to write to score full marks. Okay, because the space given to you is actually limited, only four lines, all right? So you cannot be writing everything, isn't it? If you write beyond the 4 lines, that means you have written something that's not in the mark scheme already. Okay, so that's how you roughly know whether you write correctly or not. If you feel that after you write the 4 lines, so you have not finished explaining, that means you have written something that's not in the mark scheme. Okay. Right, so next question. So this is a excitation wave that is measured in ECG, right? So you got PQRST wave according to the theory, right? okay, PQRST wave, and y-axis is electrical potential, means the voltage, and then the x-axis is the time in second. So A part 1, on an ECG, the time interval between Q and T is called contraction time. Suggest why it is given this time. So Q to T is QRST. So this is the wave that passes through the ventricle, right? So it's called contraction time because when this excitation wave passes through the ventricular wall, then the cardiac muscles at the wall immediately will contract. So contraction time because of this is ventricular systole. So the answer is ventricular systole. Okay, part 2, or you can say ventricle contract. Part 2, the interval between T of one cardiac cycle and Q of the following cardiac cycle is called the filling time. Suggest why this is given the filling time. So from T to Q, right? So from T, which is here. So T is residual wave. That means after T, there's no more excitation wave in the ventricle. After T, ventricle starts to relax already. So from T to Q, Q is when the ventricle will start to contract again. So T to Q is when ventricle totally relax. So they call this filling time. Why? Because when the ventricle relaxes, during this time, okay, two things will happen. The atrium also relaxes, right? So the blood is actually flowing into the atrium, right? From this point to this point, the blood is flowing from veins into the atria, from the two veins into the atria. So the atria is being filled with blood, okay? So sometime later after ventricle relaxes, then the AV valve will open. So when AV valve open, the blood will start to flow from atria to ventricles. All right, and then 70% of blood flow in here, remainder 30% flow in after the atrium contracts. So that's why T to Q are considered as filling time, because this is the time when the atria is being filled up by the blood from the veins and the ventricles are being filled up by the blood from atria. So that's the answer. So you need to write blood flows from veins to atria, blood flows from atria to ventricle. Okay. Right. It's already 3 o'clock. Do you have any more classes? B part 1, explain why there's a time delay between the movement of excitation wave in the walls of atria and the start of excitation wave in the walls of ventricle. So that means how come there's a delay when excitation wave move from the atrium to ventricle is of course because there's a fibrous ring or fibrous tissue or non-conducting tissue, right? Which is located between the atrial wall and the ventricular wall. Okay, so this is the first marking point. So this structure prevents the excitation wave to move directly from atrium to ventricle. Okay, so excitation wave will delay for 0.1 second at the AVN. Okay, oh no. Okay, so excitation wave will be a delay for 0.1 second at the AVN. Okay, so what else can you write to score? Hang on, let me check the mark schema. Okay. So this is the third marking point, is to prevent the overlap of atrial and ventricular systole. So that's the third marking point. Okay. All right. So part 2. Explain how this time delay helps in the heart to carry out its function efficiently. So this time delay will allow, when atria contracts, ventricle relaxes, right? So that will allow atrial contraction to push the blood into the ventricle, or it will allow the blood to fill the ventricle. okay? And then when the ventricle contracts, it will be able to push the blood into the arteries so that atria and ventricle do not contract at the same time, okay? But this mark is already given here, so therefore it will not be given in the same marking point, will not be given in 2 questions on, okay? All right, last question. Last question says that during exercise, the heart rate increases, but the contraction time normally remains almost unchanged. What must happen to the feeling time during exercise? That means to say that, let's say in a normal, when in a normal condition, this is the number of heartbeat, okay, in a certain time interval. Now, when you exercise, the heart will beat faster, right? So the interval of every heartbeat becomes shorter already. So they ask you, what must happen to the feeling time so that the heart rate can increase? Of course, the feeling time will become shorter, isn't it? So that the heart rate can increase. So the answer is it becomes shorter so that you can have got more heartbeat per minute. Okay, originally your feeling time can be very long. So now your feeling time must be shorter already. That means in the entire cardiac cycle, certain timing is for the feeling time, okay, of the atria and the ventricle. Certain timing is the ventricular systole, okay? So the feeling time must be shortened. All right, so that's all for today.